Well, hey guys, Mrs. Shelton here. This is our last unit review, Unit 6, Gene Expression and Regulation. So with this particular unit, this is your test prep review book, topic 5. So if you're actually using that test prep review book, you only need to go through the first half of it because Unit 7, Unit 8 is not directly covered on this year's AP exam. Do keep in mind, even though natural selection and ecology are not directly covered on this year's exam, they are embedded in those big ideas. So big idea number 1 on evolution covers all units 1 through 6. Big idea number 4, interactions, covers all units 1 through 6. So even though it's not directly going to be asked, make sure you're keeping in mind what are some of the evidences of evolution, or what are some of these interactions that we talk about. So even in gene expression and regulation, we'll address some of those. Now, topic 5, definitely make sure that you are understanding that, but you can also see that it's a lot of textbook chapters, so 13, 14, 15, 16, 17. So this is why we're just spending one full week on Unit 6 review. Now, this Screencastify, if you are using these Screencastifys to help you review, I do not expect that you are going to be able to sit through this entire Screencastify because there's a lot of content. Unless you just play it in the background and you fall asleep to it. So maybe cut it off halfway through, and that's a-okay. And then watch the other half later on in the week in order to help you guys focus. Now, what College Board tells us as far as developing our understanding, we need to be able to compare DNA and RNA. This should be something that's very easy, but it really is about understanding this protein synthesis, understanding transcription and translation, and also this regulation of gene expression. How is transcription regulated? And then what are all of the different regulation mechanisms that do occur that lead to cell specialization? Now, in our preparing for the AP exam content, here are some things to just make sure that you are seeing in here. When we talk about gene expression, they are directly telling us that we need to make sure that we are understanding how the lac operon in E. coli is an example of this positive gene regulation. Also, they're telling us in here about common errors of mutation. And so what students often think is that mutation is bad. And we have discussed ad nauseum in class that most mutations end up being neutral mutations. But it really is up to natural selection to determine if a mutation that does cause a change in a protein that changes the phenotype is positive or negative. So it's recognizing all of those different examples of mutations and not getting stuck in that idea that a mutation is bad. Now, with our big idea questions for this, the big ideas here, they're just focusing on big idea number 3, because that's what this whole unit really focuses a lot on. But we do need to make sure that we are recognizing the energy part from big idea number 2. Building proteins requires a tremendous amount of energy. When we talk about big idea number 1, we need to make sure that we recognize that DNA and RNA is a part of all living things. When we're talking about transcription and translation, there's some slight differences that exist between prokaryotes and eukaryotes, but it's all the same enzymes. I mean, there's DNA polymerase, there's RNA polymerase, and these are all conserved events. And then here, when we're talking about just gene regulation, there is differences, though, in gene regulation to make sure that we are recognizing how genes get regulated in eukaryotes versus prokaryotes is what we should make sure that we are identifying. And then how this genetic information, how does it get passed from generation to generation? And this is tying into unit number 5 on heredity as well. Now, here's our unit at a glance. So you can see that there are 8 different topics that we are covering here. So just basic DNA and RNA structure, we're going to talk about DNA replication, and then we're going to talk about transcription, and then RNA processing in eukaryotes, the translation process, and then we'll talk more about this regulation of genes. So gene regulation in prokaryotes and eukaryotes, and also then this gene expression and how that leads to cell specialization. Types of mutations we need to make sure that we're reviewing, and then we also need to review a little bit about biotechnology. So let's go ahead and begin. Let's move me out of the way here. So with this diagram, everybody should be recognizing this diagram here as our central dogma of information flow in living things. DNA is the genetic material for most living things. And when I say for most, RNA sometimes does carry the genetic material, but that's more common in viruses that are not living, where RNA is the genetic material that gets passed in a virus. But from DNA, DNA is the instructions for making the RNA. When we talk about RNAs, we have to recognize that all RNA was actually built from the DNA. And then it's through RNA that through the translation process, then proteins are made. So we need to make sure that we are very, very clear on how that occurs. Now, when we talk basic DNA, RNA structure, this is all stuff that should be very, very familiar to you. We need to first recognize that with DNA and RNA, or nucleic acids, the monomer is a nucleotide. Nucleotides consist of three parts. It's a phosphate, a pentose sugar, and a nitrogenous base. Now, obviously, the differences between them are the pentose sugar. Here, this is a deoxyribose because there's no oxygen down here. So this is the pentose sugar of DNA. If this was RNA, I would have another hydroxyl group down here, and that would be my ribose sugar that I would be finding. As far as my nitrogenous bases, remember that in DNA, I have 4 nitrogenous bases. I have G, C, A, and T, guanine, cytosine, adenine, and thymine, so GCAT. And those nitrogenous bases are purines or pyrimidines. So remember our mnemonic, GCAT, puppy, puppy, 32. In DNA and RNA, G and C are complementary bases. Guanine is a purine. A purine is a double ring structure, and a pyrimidine is a single ring structure. And so remember, poo is bigger than P, so that's your easy way to make sure that you're remembering that. But also then guanine and cytosine are going to make 3 hydrogen bonds, and that's what holds the 2 strands of nucleic acids together, are these hydrogen bonds between these nitrogenous bases. So making sure that you're aware of that. Now, if we were making a mnemonic for RNA, it would not read GCAT, it would read GCow. There's no T's in RNA, there is uracil instead. And uracil, though, is that single ring structure that is your pyrimidine. So here is your overview diagram, making sure that you're recognizing that DNA is a double helix. Being a double helix, it is 2 polynucleotides. Remember, this diagram isn't showing it, but remember that these are anti-parallel. And so with anti-parallel, that's going to be important anytime that we use the template strand of DNA to now make RNA from it. Remember that the complementary bases have to go anti-parallel upside down versus the other. How we know which direction it is we do have to look at our nucleotide at the end in order to determine if it's the 5' end or if it's the 3' end. So if this end up here was the phosphate group that was sticking out, remember that's going to be my 5' end of that polynucleotide. That means the other side, what's going to be sticking out is now this hydroxyl group. So that's going to be my 3' end. And remember the 5' and 3' are related to which carbon is this functional group attached to. This would be my 3rd carbon of my pentose sugar. This is my 5th carbon of my pentose sugar. But you do need to remember phosphate 5. It's kind of easy to remember it that way. So basic DNA, RNA structure, and structural differences, incredibly important. And they also specify, make sure that you do know purines versus pyrimidines and which is which of our nitrogenous bases. Now, what you should also recognize, because this is going to play a role in our understanding of gel electrophoresis, your phosphate group has a negative charge to it. And so what do we know about charges? As opposites attract. And so that's the basics of gel electrophoresis in a gel box is our nucleic acid fragments are going to move towards a positive charge. So with DNA replication, so this is our next concept here, is DNA replication, DNA copying itself. And this is how we have to make sure that every cell gets a new copy of its genetic information. So it's information for making proteins. So with replication, replication is very similar in all living things. And with that, what we have to make sure that we are recognizing is that DNA does get synthesized in the 5' to 3' direction. And so it's very important that you know that new strands of DNA get built 5' to 3'. And so that's going to be the role of our DNA polymerase. Now in here, they're telling you which enzymes for replication you need to be familiar with. So we need to recognize overall replication is always semi-conservative. That means we're always going to save one strand of old DNA and build a complementary strand on top of that. Enzymes we need to know, helicase for unwinding the DNA. We need to know topoisomerase for unwinding upstream. We need to know DNA polymerase is what is going to be elongating the new strand of DNA 5' to 3'. But we also have to make sure that we're recognizing the role of RNA primers. DNA polymerase can't just start making new DNA unless there's an RNA primer that was laid down by primase beforehand. We need to make sure that with DNA polymerase, recognize here, they don't care if you know the difference between DNA polymerase 3 versus DNA polymerase 1. So we learned that in class, but College Board does not need you to know that. They do need you to know ligase though, and making sure that you recognize the role of ligase in joining those fragments together on that lagging strand, those Okazaki fragments on that lagging strand, because that's not something that polymerase can do. And also remember too, I mean, that was one of the roles of your DNA polymerase 1 was to remove RNA primers, but it couldn't then connect the new fragments of DNA together. So we need this ligase in order to do that. So here's a diagram that you feel very, very familiar with. You should feel familiar with. This comes from our Campbell textbook. And so with this, remember I've got my replication fork here. And with this, here's my origin of replication. This red is representing an RNA primer that was laid down. So my DNA polymerase can build DNA in that 5' to 3' direction. DNA polymerase can only attach nucleotides to a 3' end. So it builds continuously in a 5' to 3' direction. This is why at every replication fork, here's my helicase that's unwinding. And so at my replication fork, I'm going to have one strand where the DNA polymerase can continuously create a new strand of DNA. But in order to keep up with helicase, the other side, because it can only build 5' to 3', is now going to be making its DNA in fragments. And so that's why I have a leading strand and a lagging strand. And the reason I have a leading strand and a lagging strand is because DNA polymerase can only elongate DNA in the 5' to 3' direction. So make sure you're remembering that. Now, this diagram shows you the role of ligase here in joining the fragments. It's creating this covalent bond. Here's my DNA polymerase 1. Again, you don't have to know the difference between 1 and 3. So this is actually removing the primers and replacing it with DNA nucleotides. And then my DNA polymerase 3, again, is building 5' to 3'. Now, primase that you see here, this again is not an enzyme you have to know for the test, but we do need to know that primers are necessary, and these primers are RNA primers that will initiate the replication process. Now, I gave you this diagram because the last diagram just didn't show the topoisomerase. So this is the exact same diagram that we just saw, except here you're seeing the role of topoisomerase that's working upstream. So here's my helicase that is breaking the hydrogen bonds between the two strands. Again, we're seeing the semi-conservative nature of replication. And my topoisomerase is helping to unwind the DNA upstream before helicase gets there. Now, remember that DNA replication. When we talk about DNA replication, remember that this in eukaryotes happens during the S phase, is what is happening. So this is way before mitosis and cytokinesis ever happens. This is all happening during that S phase of the cell cycle. Okay, so concept number 3, transcription and RNA processing. So let's take a look here at some of the learning objectives and essential knowledge before we start talking about it. So when we talk about protein synthesis, so remember with protein synthesis, we start with transcription first. Transcription is that DNA to RNA, to messenger RNA, and then translation that happens with the ribosome is my messenger RNA to my polypeptide. We have to make sure that we are aware of our 3 different types of RNA that are involved in transcription and translation. So we need to make sure that we recognize our messenger RNA. Messenger RNA is a copy of the gene, so in RNA form, so it's a copy of the template strand of DNA. My ribosomal RNA, my ribosomes have 2 subunits. So there's an upper large subunit, there's a lower small subunit. These are made up of ribosomal RNA. My rRNAs are going to be where protein synthesis actually happens, where translation occurs. And then I'm also going to have my transfer RNA, my tRNA, is going to be the strand of RNA that has 3 bases on here that are called anticodons. that are going to be complementary to the codons on messenger RNA. Remember, transfer RNA's role is to carry the amino acids to the growing polypeptide that's being built at the RNA. So with this, we do need to also remember here just some other terms they're talking about is we need to make sure that we're recognizing the role of an RNA polymerase as well, and making sure that we understand that that's the only enzyme that is needed in order to initiate this transcription process. And we'll talk about the control of it, and we need transcription factors and enhancers and those things. But this is a one-stop shop where RNA polymerase can actually unzip the DNA, and it can actually make a copy of the DNA off of the template strand. So it's always the template strand that the messenger RNA is being built off of. Now, RNA polymerases, they also can only build RNA in the 5' to 3' direction. So it can attach a new RNA nucleotide to this 3' end and elongate it that direction. Now, also too, when we read our messenger RNA that has codons, we read messenger RNA in the 5' to 3' direction too. So remember, I already told you, in AP Bio, when in doubt, everything always goes downhill is what we need to make sure that we are addressing. Now, also when we look at DNA, since DNA being this double helix, this is my template strand. That template strand also gets referred to as the non-coding strand. So don't get confused if they don't use the term template strand. The other side is referred to as cDNA or the coding strand of DNA, and they call it that because it's going to have a sequence that is almost identical to the messenger RNA, because remember complementary base rules. So except my coding strand is going to have T's or my messenger RNA is going to have U's. But you will most often be given the template strand, and then you have to use the complementary base pairing rules of GCAT, GCow in order to make sure that you know then what that messenger RNA is, and then be able to use the universal genetic code to determine what amino acids is that coding for. But do keep in mind, template strand is what messenger RNA gets built off of, and then the other side of DNA that is not getting transcribed is referred to as the coding strand. Now, do remember here, so this is the continuation of the concept. So they're talking about RNA polymerase synthesizing in the 5' to 3' strand. Up here, they're actually talking about the fact that the template strand is also referred to as the non-coding strand. Sometimes it's referred to as a minus strand or an antisense strand. Most often, though, it's going to be referred to as template strand or non-coding strand. And the other side of DNA is going to be referred to as the coding strand. So and that's where you see it here in this particular diagram as well as the template strand versus the coding strand. Now, what we have to also make sure that we are recognizing is that in eukaryote cells, eukaryotes have now my RNA processing. And in RNA processing, what's going to happen is there's going to be an addition of the poly A tail, there's going to be a 5' cap, and then we're going to be cutting out the introns in this splicing and understanding alternative splicing as well. So remember that in RNA processing, after that primary transcript is copied, so this is my messenger RNA up here, I'm going to have sequences that are classified as exons or introns. Now, during RNA processing, this is going to happen inside the nucleus, is where RNA processing takes place. Introns get removed. So you have to make sure that you are remembering that. And maybe during the test, if they show exons and introns and you're not remembering quickly, just look it up in your notes and make sure you remember introns are the ones that get cut out. And so those get spliced out and connected back together. And then we have a 5' cap that is added and a poly A tail. So if you ever see messenger RNA that has a 5' cap and a poly A tail, you are also going to know that this is in a eukaryote cell is what we are addressing. Now, the reason for this splicing, and this is again, here's another example of the role of RNAs, because remember we had these little SNRPs. So these SNRPs and proteins that create this large spliceosome, this RNA enzyme complex, this is what is removing these introns, is what is occurring here in this process. But the reason for this, and this is really important that you guys understand, is what makes this so advantageous to eukaryotes is that in one gene, I can actually have multiple variations of proteins that are produced from that one gene through alternative splicing. And so this is an example just showing you some alternative splicing. And this is just using these 5 exons. So these exons being the coding segments. Well, here in alternative splicing, here I use all 5 in this sequence, but here I'm only going to be using 4 of them. And here I use a different 4. Well, that results in some different protein structures that then have some different functions. So keep that in mind and why that's such an advantage of one gene can have multiple different proteins that are produced from that one gene due to alternative splicing in eukaryotes. Now, concept 4 is now the translation process. And in the translation process, one of the differences you should know between prokaryotes versus eukaryotes is that when transcription is happening in a prokaryote, translation actually just starts right on top of it. So I have my ribosomes that attach at the start codon, and they just start translating and reading those codons and turning them into a polypeptide right at that same time, right in that same space. But because eukaryotes are compartmentalized, I'm going to have my transcription and my RNA processing happen in the nucleus, but now I'm going to have my messenger RNA go out into the cytoplasm. So whether they are bound or they're going to be free ribosomes. If they're bound ribosomes, they're on the rough ER, and that's where most of our protein synthesis is going to take place. If it's a protein that's intended to stay inside the cell, then this messenger RNA might attach to free ribosomes that are not associated with the endoplasmic reticulum. So make sure that you're aware of just that difference of where does translation occur in a prokaryote versus a eukaryote. So when we're talking about translation, this is now that messenger RNA that is going to attach to the ribosome. The ribosome is going to initiate this translation in order to now be able to translate it into a polypeptide. So with this, how this initiates, and this is what we need to make sure that we are recognizing, is that this process of translation, so here's my messenger RNA in a 5' to 3' direction, here's my RNA, here's my tRNA. Well, what's going to be happening is the initiation is what is going to start this process, and I can have a whole bunch of sequences up here starting at my 5' cap that mean nothing. My ribosome is only going to attach where I have this AUG start codon that codes for the amino acid methionine. So it's this MET start codon, and this is where that initiation is going to occur. I'm going to have my tRNA with its anticodon attached to the codon, and then my 2 ribosomal subunits are going to attach, and then my ribosome is just going to now, just like little machine, keep on zipping through, and as it zips through, remember that with this, I have 3 different sites in my ribosome. College Board does not need you guys to know the E site where the tRNAs exit, the P site where the growing polypeptide is, and the A site where tRNAs attach. But you should understand that this is a process of each tRNA bringing in the amino acid, matching its anticodon to the codon. And so termination, this is going to continue until we hit a stop codon or a termination sequence. And so at that termination, when a stop codon occurs, again, it doesn't matter if there's still thousands of nitrogenous bases that still exist. As soon as there's a stop codon, there's a release factor that binds to that A site, and that is going to cause the ribosome to detach, and that's going to cause the completed polypeptide to also detach, and then it's going to continue to get reformed. Because remember, this is just the primary structure that is being formed right here of the polypeptide. So it still has to go through folding processes for its secondary level, tertiary level, and potentially quaternary level. And that's all going to happen then through that endomembranous system. So from the rough ER, then it's also going to be through the Golgi complex where modifications can happen. So just keep that in mind. We are simply talking about how do we go from the nucleic acid to now we're going to the polypeptide. Keep in mind too, where this ties in with energy. We are building this long polypeptide, this movement of molecules, this attachment of an amino acid to tRNAs. Those are all processes that require energy to do that. So keep in mind, this whole thing is a huge, huge energy using process. When we do talk about this, the last part, we addressed all of this essential knowledge up here as far as tRNAs and and messenger RNAs and our amino acids. But we also need to recognize the role of retroviruses in as a particular biological agent that goes against the central dogma of information flow. So we definitely need to make sure that we are addressing that. Um, we need to make sure that we are addressing the universal genetic code, the fact that the universal genetic code, remember the universal genetic code is written in messenger RNA. And so this is messenger RNA written in a 5' to 3' direction. And it's imperative to make sure that you know that these messenger RNA codons, remember that wobble effect, that oftentimes that third base in a messenger RNA does not change the amino acid. And so that's going to play a big role in a lot of our neutral point mutations. So these point mutations that end up just being neutral, not resulting in a change in the polypeptide. If there's no change in the polypeptide, then there's no change in the phenotype of the organism. So with this, remember that since the majority of life on this planet uses the same universal genetic code, this again is evidence of unity, of a common ancestor that has similar processes. Now, retroviruses, why we need to make sure that we are recognizing retroviruses is because there are cases of living things or biological agents going against this normal flow of information from DNA to RNA through that transcription process, RNA then to my protein or polypeptide through the translation process. A retrovirus is an RNA-based virus, and not all RNA-based viruses are retroviruses. Like COVID-19 is an RNA-based virus, but it's not a retrovirus. This happens to be in something like HIV. HIV is your example of a retrovirus where its RNA is its genetic material, but it carries with it this enzyme called reverse transcriptase. So if it's reverse transcriptase, remember ASE, it's an enzyme. So this is something that does reverse transcription. So it allows for RNA to now be used as a template to now form DNA. And this DNA now becomes a provirus in the host cell. And now that DNA now can be used to now make more virus. Okay, concept 5, regulation of gene expression. So with regulation of gene expression, because we know that we have trillions of cells in multicellular organisms in us, but they're all different. And what is different is what genes got expressed and what quantity of those genes got expressed as far as the proteins being produced. So we need to make sure we're understanding that there are regulatory sequences in DNA that are going to help control gene expression. We also need to know this word, epigenetic changes, because it's not just did you get the gene on whether or not it gets expressed. There are some environmental factors that can influence gene expression as well. So we need to make sure that we're understanding that through these modifications of histones. So we'll talk about that methylation process. And then we need to make sure that we are understanding down here this inducible or this the control of gene expression in prokaryotes versus eukaryotes, and especially we need to refamiliarize ourselves with the lac operon, so in a prokaryote. Now, if I'm looking here, you should all be able to look at this and instantly and immediately recognize that this is eukaryote gene control. So, and how you would know this is because of this enhancer region of the DNA, these activators, and then I also have my transcription factors that are all going to be landing on this promoter region. Now, both prokaryotes and eukaryotes have a promoter region, and so this is a sequence in the DNA that is upstream from the gene. And so here's my gene that is going to code for the polypeptide. This is what is going to get transcribed and then hopefully translated if it's not blocked. But remember in promoter regions always have these random sequences of T's and A's. So it gets referred to as a TATA box. So if you ever see a TATA box, you know you're talking about a promoter region. But from this control, I have general transcription factors that actually attach at the promoter region. And then I also have activators, these other proteins or distal control elements that attach the enhancers. And we get this bending that ends up occurring that now enhances and tells RNA polymerase this is where we begin transcription. So this is eukaryote control. In prokaryotes, this is my lac operon. And remember that in my lac operon here, what I'm going to actually have for the breakdown of lactose, the disaccharide lactose, that milk sugar. For this to happen, there's actually 3 enzymes that are necessary to break down that disaccharide lactose in order to convert it into a usable sugar for prokaryotes. Now, lactose is not always present. So there's no sense in making these enzymes to break down lactose unless lactose happens to be present. So one of the things to be aware of is that with prokaryotes, they're very efficient in their regulation of gene expression. They put all enzymes for the same biochemical pathway all together with one promoter region and one operator region. And so if I activate this particular promoter region and allow RNA polymerase to transcribe, well, then all 3 of these genes are going to get transcribed all at the exact same time. Now, since lactose, this operon then is inducible because lactose is not always present. There is a gene that actually codes for a repressor. There is a repressor protein, and this is my repressor protein that was coded by another gene that would attach right up here at this operator region and block RNA polymerase from being able to transcribe the rest of these genes. However, when lactose is present, lactose being this environmental inducer, lactose binds to the repressor and inactivates it, and then it falls off, and now RNA polymerase can transcribe these enzymes, and now the prokaryote can use that lactose as an energy source. So this would be this inducible type of operon. So make sure you're familiar with the lac operon. College Board specifically says lac operon several different times throughout the CED. Now, when we talk about epigenetics, when we say epigenetics and these factors, epigenetic mechanisms that are affecting gene expression, these are a lot of these environmental factors. And really what's happening in any environmental epigenetic factor that's different. So it can be drugs, pharmaceutical, diet, it can be in utero. Any of these, what's happening is it's affecting the chromatin. And so we talked about DNA methylation. And so with DNA methylation, remember if this DNA gets methylated here at the histone, remember histones are these proteins and DNA winds around it in order to become ordered. Well, with methylation, that actually binds up the DNA and makes those segments inactive and transcription can't happen off of this DNA. But also remember too that we also refer to as the acetylation process. There's other epigenetic factors that actually open up these histones. And now with that modification now allows for that DNA to be accessed and that gene can now be transcribed. So we talked about DNA methylation. That can be an epigenetic mechanism. And we also talked about that histone acetylation that also opens up that DNA that can also be an epigenetic factor too. So the big key here is that you are recognizing that environment can also play an influence over gene expression. And this is just through that structure of the chromatin. Is it that heterochromatin or that euchromatin that we talked about? College Board doesn't need you to know those terms. They just need you to know, is my chromatin tightly packed? If it's methylated, then these segments are off. We can't access that DNA. And if now it's unpacked, well, now my DNA is accessible. When we talk about 6.6, gene expression and cell specialization, this really is now just making sure that you are understanding how we get cell differentiation, cell specialization in multicellular organisms. And it's all about gene expression. Which genes got expressed and what quantity of proteins got expressed and in what manner, because it's all about the proteins that make a cell different. Now, here again, we're talking about regulation of gene expression. We're talking about transcription factors again. We're talking about promoter regions again. We are talking again in here about now are small RNA molecules that have roles in regulating gene expression. So you should have in the back of your head some idea of some different roles RNAs have other than just messenger RNA, rRNA, and tRNA, ribosomal RNA, and transfer RNA. We should also know our siRNAs. You should maybe know your SNRPs for excising out exons. But when we talk about cell specialization, it really is understanding that from a single cell, it really just depends on which genes are going to be activated to produce those proteins to now differentiate between a different cell. I mean, remember, you all started out as one single cell, but those cells had to take on specialization. We also have, and it's not just the initiation of transcription that controls it, there are also repressors, even in, and we're seeing this folding here, so you should recognize that this is a eukaryote that we're talking about. There are repressors that can actually bind in order to stop that transcription from happening too. This particular diagram is actually now just helping you to remember the role of some of these little small RNAs that actually, with their anti-parallel sequence, can actually attach and cause the degradation of messenger RNA that got translated. So we can have translation happen, but now we can have these little small RNAs that actually break that RNA down, or they can actually bind to, we can have some other RNAs that actually bind and stop that translation stop that translation from happening and block that ribosome from ever attaching. This is a modification of a diagram that we talked about in our book that talks about all of the different levels of gene expression control in a eukaryote. And really, at every level, eukaryotes can control this ultimate gene expression. And so it can be in the packing of chromatin that we just talked about, where we talked about our heterochromatin or our euchromatin, whether it's open or closed at the histone level. Initiation of transcription is where it can happen. Our RNA processing is where we can control what polypeptides are there. We can also have those little small RNAs that actually break down the messenger RNA that was produced. In translation, we can actually block translation from happening. The polypeptide can also, through enzymes, be broken down. And then there's also these post-translational modifications, these folding of these proteins that can also change too. So all of these different levels of protein synthesis, from just structure of our chromosome to the ultimate polypeptide, these are all different ways that we can affect that ultimate protein shape, or no shape at all, or no protein at all, that it's the proteins that now give us phenotypes. And so that's really what we talk about when we talk about the phenotype, is what protein conformation is formed in order to now give us that overall function of a cell. And remember that cells affect tissues, tissues affect organs, organs affect organisms, and everything is interconnected. And then it's up to natural selection to choose, is this a good shape of a protein or a good production of a protein to increase reproductive success and survival. All right, 6.7 on mutations. So this should be a review. Nothing new here that should freak you guys out as far as mutations. Remember, mutations happen. Mutations are the primary source of change in DNA, and DNA changes. Mutations happen. You are all mutants. You have mutations, but most mutations end up being neutral. Remember that when we have DNA mutations, they can be positive, they can be negative, or they can be neutral, depending on their effect on the organism and in natural selection. So with that, we have to keep that in mind. But we also have to make sure that we are clear that mutations can either be at the point level, so just the nucleotide level, but we can also have some large chromosomal mutations as well. Remember that rate during DNA replication, and DNA polymerase is not exact, and also the proofreading is not exact. There's also different mutagens that occur, and mutagens that are environmental exposure that can also change DNA sequences. And there's all sorts of effects or no effects whatsoever that these mutations can actually cause. So again, talking more about mutation, but in this segment though, what's really important that you pay attention to, and I'll give you a slide, is making sure you understand how prokaryotes, other than just changes during replication, prokaryotes actually have a wide variety of ways to take in new DNA to get these genetic changes. So we need to make sure that we are recognizing this, also recognizing how viruses mutate as well. So these would be some points to make sure that you are remembering, not just our different types of mutations that are kind of common. So this is actually with our point mutations, this is something that I would assume that you guys are very comfortable with. When we talk about our point mutations, these changes in just one single nucleotide, so one change in the letter. And so here's my original DNA level. And so now here's my messenger RNA. But now if my DNA had this one change in this third letter of a codon, this would be a silent mutation because this change in the codon didn't change the amino acid that was put down during translation. So you're not going to be able to tell in the phenotype that any type of mutation occurred. Now, we can also have point mutations that lead to nonsense. And this nonsense, so in this case, I can have a stop code or a start code that's actually put in the wrong place. So now I either don't get the same polypeptide, I don't get a polypeptide at all, or I get a completely new polypeptide. And that's my nonsense point mutation because of now changes in that stop or that start code. I also then, this is the most common, is now a missense. And in a missense, now I have this one base substitution that now results in a new amino acid in this polypeptide. And so this one change in amino acid can actually change then how that polypeptide behaves in different environments. And we'll take a look at this when we talk about sickle cell. Now remember, frameshift mutations. Frameshift mutations happen if there is a substitution or a deletion. And if it's a substitution or a deletion, it's a substitution or deletion of one or two bases. If it's a substitution of three, remember we had a test question on that, then that removes one full codon. And if we remove one full codon, it's not a frameshift. It's only a frameshift if now every single codon is affected after that mutation. Now, we talked about chromosomal mutations that can happen too. And so during mitosis and meiosis, now we can actually have sometimes full pieces of chromosomes that actually change. We can have deletions, and then we can also have duplications where segments get duplicated. We can have inversions where segments get flipped upside down. We can actually have translocations that are occurring. This is also where we have polyploidy or aneuploidy, where we have missing or extra chromosomes as well. So recognize all different types of mutations at the chromosomal level that can happen during mitosis and meiosis or at the nucleotide level. Here's just trying to show you during normal cell division. This is something you should recognize is if I have non-disjunction during meiosis 1. If the non-disjunction event happens during meiosis 1, then all of my gametes are going to be affected in their chromosome number. If I have non-disjunction occurring in my second round of cell division, then I only have two gametes that are affected and I have two gametes that are normal. So pay attention to that. That's an example problem that College Board gives you. It's a previous test question that they gave you too on can you recognize when the non-disjunction during meiosis happened? And always look at the result because if I have non-disjunction during anaphase 1, then all four gametes are going to be affected. Again, if I have non-disjunction during anaphase 2, then I only have two gametes affected and I have two that are normal. So pay attention to that difference. This particular diagram is just showing you some different mutagens. And so these would be some different causes of mutation from UV radiation and X-rays to different chemicals to infectious agents like HPV, a virus that becomes a provirus. It's inserting its genetic material into your genetic material. So therefore, these are all causes of mutation that can cause them. Now, here is what I need you guys to make sure that you are writing down and you're remembering with prokaryotes and how prokaryotes can acquire new DNA in addition to just their mutations that happen during replication. So first of all, we have this term conjugation. We had talked about the fact that DNA can exchange these small little circular plasmids. And so with this, they will actually exchange, it's called a sexpilus, and they can actually pass on plasmids to other bacteria. So this is conjugation is one way to exchange or to obtain new genetic material. Transduction, here's my bacteriophage. Bacteriophages will often insert their DNA into a prokaryote DNA. So this becomes, instead of a provirus where we talk about in a eukaryote, this becomes a prophage. And that prophage then is new DNA inserted into the genome of that prokaryote. And then we also have natural transformation. And so through that natural transformation, prokaryotes will actually, during times of crisis, when they become competent, is what it's referred to, they will start uptaking some of their surrounding dead relatives' DNA and start uptaking some of that DNA. Just why not see what happens? I mean, really, that's all mutation and evolution really is. It's all luck of the draw. Does it work? Here's another source of genetic change is transposons. Remember we talked about these jumping genes. Transposons are transposable elements that can actually now move to different locations, which can now also change that production. In viruses, viruses mutate all the time. This is why coronaviruses, there's a wide variety of different coronaviruses. COVID-19 is a new form of coronavirus. It's an RNA-based virus. But in viruses, remember when a virus does inject its genetic material, it's using that host cell to now produce all of the protein capsids and also all of the genetic material and then reassembling. Through that reassembling process, sometimes just even during reassembly or even through that making of the new genetic material, there's mutations, there are new arrangements that happen. Now, if you get co-infection, so if you had two viruses that actually infected the same cell at the same time during this reassembly process, then you can get new combinations of genes that reassemble to create a new form or a new strain of the virus. Now, again, mutations, we don't know if they're good, bad, or neutral until natural selection acts upon them. And it's definitely neutral if we see absolutely no change in protein production. However, if it is a positive mutation or a negative mutation, it just depends on natural selection. Does it increase that organism's chance of survival and reproduction? Then we can tell if it is positive or negative. That's the ultimate test. Some illustrative examples. One of the illustrative examples that we have of mutations, we talked about the cystic fibrosis gene. We did this in another unit. We also did it in class. So the CFTR gene that now changes that chloride channel. And so that leads to now this effect on the entire organism. Another example that we have that we did not talk about in class. But again, illustrative examples, you guys don't need to know all of them, but here's actually a change in what's called this MC1R gene. And so with this MC1R gene, this mutation actually now allows this particular mouse to actually start, it changes its coat color depending on the soil color, depending on its environment, based on this mutation that happened in this melanocyte stimulating hormone that's here. Here's an illustrative example that we did talk about. We talked about antibiotic resistance. Remember that in antibiotic resistance, this mutation happened in the DNA, and the variation has to already exist in the population. So it's not that the use of antibiotics caused the mutation. The variation had to exist, but this was a mutation that wasn't selected against, for or against, because it wasn't necessary in the environment until the environment changed. And when the environment changed to now be around antibiotics, having this mutation now led to antibiotic resistance. And so these particular prokaryotes survive. Well, now you keep that environment the way it is. Well, these are the guys that keep on reproducing. And so now we have a whole colony of prokaryotes that are all resistant to that particular antibiotic. Exact same concept happens in pesticide resistance. The change, so the resistance gene had to already exist in the gene pool. So it was a mutation that wasn't selected for or against until the environment changed of now having this pesticide. And so now you have some pests that are resistant to that particular pesticide. Those are the ones that now survive in that environment and continue to reproduce. And this example of the sickle cell disorder and heterozygote advantage, we have talked about a lot. I've had you guys do a lot of work with it. This is just a classic example of a point mutation that resulted in a missense. And so with this, I have one change in amino acid. And so that change in amino acid that now changes the hemoglobin protein when your blood becomes more acidic due to lack of oxygen and buildup of carbon dioxide. Well, this is going to change the shape of that particular protein. And so if you are homozygous recessive, you have sickle cell disease. But if you are heterozygous, meaning that you have one gene that codes for normal hemoglobin, one gene that codes for the sickle cell, you are actually resistant to malaria. And last but not least, we're finally almost done. I know this is a really, really long section. Again, this is why I'm having you guys review it over the course of an entire week and not 2 units to cover. Biotechnology, we actually covered more in class about biotechnology than what College Board needs you to know. Again, I try to over-prepare you guys for everything. But with these engineering techniques, what we understand about DNA replication, what we understand about the properties of DNA and RNA, we can use in lab practices. College Board needs you to understand gel electrophoresis and why and how it separates molecules according to size and charge. Needs you to understand polymerase chain reactions. PCR is for amplifying DNA fragments and actually RNA fragments. Also bacterial transformation. So when we are creating transgenic organisms, so this bacterial transformation when we're inserting DNA into bacterial cells, and then also just kind of the basics of DNA sequencing in order to understand what is that sequence of DNA as well. So in gel electrophoresis, we talked about here with gel electrophoresis with your DNA is cut into fragments. And so those fragments, so using a specific restriction enzyme are going to create the fragments. And so then through the gel box, you allow the gel to run and pull these fragments towards the positive end. And so what's going to happen is, remember, the more gelodynamic fragments, these smaller fragments can get through this gel at a faster rate than the larger fragments. So it separates these fragments based on size. There is a log graph paper that you can use in order to figure out exactly how many nucleotides each one is. You do not have to do that. Maybe in your college class, they'll make you do it once just so you have seen it. Remember in PCR, the purpose of PCR is to make copies of DNA. You get a small little DNA sample, and so in a lab, we can make copies of that DNA or even that RNA. That's actually part of COVID-19 testing right now is PCR. They're taking samples from the sinus cavity, and so with that, then they are trying to use PCR in order to amplify the RNA fragments in order to then be able to test it. And that's kind of what some of the backlog is and some of the testing for COVID-19 is the lack of PCR capability and then some of these buffers and solutions that are necessary. So what you need, though, if we're going to be replicating nucleic acids, well, we have to have primers to initiate that. We need to have free nucleotides as well. And we also have to have the enzymes that are going to do the replication process. When we talk about transformation of prokaryotes or bacteria, so we did talk about the transformation activity, Griffith's dead mouse experiment, where he determined that there was some type of molecule that was a transforming agent. Well, we can transform bacteria by genetically engineering a plasmid, these circular pieces of DNA that bacteria readily will change. We will use an endonuclease, a restriction enzyme, in order to cut it out, creating our sticky ends as well, and inserting and creating this transgenic piece of DNA. But now in order to get bacteria to uptake that is we force this bacteria cell to become competent. And in order to force it, we just change the environment very rapidly. We can either heat shock it or we can cold shock it. We don't want to heat shock it to kill it, but we can do just a dramatic environmental shift. and that'll make it actually want to take in other sources of DNA and try to transform that cell. And so now if it has new genetic material, now it creates new proteins, and so now we say that that cell has been transformed. If we are talking about sequencing, in sequencing, they used to use PCR, but they would use now radioactively tagged nucleotides. And so with that, with PCR, then they would separate after PCR, then they would actually separate fragments based on size, and then based on where those radioactive tags ended up, then they could actually sequence it. It was a long, long, tedious process when it was done by hand. Now sequencing is all done by computer and computer technology, so it does not have to be done in this particular way anymore, but it still is a pretty complex process that now computers do for us instead of by hand. Our illustrative examples, when they say amplifying DNA fragments, remember that they are talking about polymerase chain reaction, PCR, replication in a test tube. When we are talking about analysis of DNA for forensic identification, well, this would be using gel electrophoresis to create a DNA fingerprint and seeing then if the DNA fingerprint of suspects then match the way these fragments are separated out. Because again, where restriction enzymes cut my DNA is going to be different from where restriction enzymes cut your DNA. When we talk about our next example here, let's go, boop. When we talk about genetically modified organisms, include transgenic animals. If we say it's genetically modified, that means that it has DNA from two different sources in it, is genetically modified. So we have altered the genetic makeup of that particular organism. Now, if it's a transgenic animals then are going to have genes from another animal in them, is what that means. And then gene cloning, when we talk about our gene cloning, this is our basic bacterial transformation where we have our host gene. That host gene is cut out with a restriction enzyme. Here we have a plasmid that's also cut with the same restriction enzyme. Now we insert, we create this recombinant DNA, and this recombinant DNA, then we can make this bacterial cell competent, so we can heat shock it or cold shock it in order to have it uptake this plasmid, and then now it is a transgenic bacteria that will replicate because prokaryotes replicate asexually. And so that's a way to clone then this gene that we put into this plasmid. So this would be an example of a transgenic bacteria with its recombinant DNA. Genetically modified organisms have recombinant DNA. Transgenic animals have recombinant DNA, what they have. So that is all of Unit 6. When we have our review session on Unit 6, we will go over some of these practice FRQ questions, and then you will also do the practice FRQ questions from previous AP exams that you're going to be turning in. But with that, if you stuck with me through this entire unit or you broke it into 2 days like I suggested, that is awesome. And I will talk to you guys soon. I miss you. Wish we were in class so we could go over this and I could answer your questions directly. But good luck studying, and I will be in contact with you guys soon.